Welcome to Nasdaq Trade Talks. I'm Jill Malandrino, Global Markets Reporter at Nasdaq. Joining us is Mark Emalfarb. He's the founder and CEO of Dyadic International. We're going to learn how the C-1 gene expression platform can potentially make COVID-19 treatment more affordable. Mark, it's great to have you back with us on Trade Talks. Thank you so much for joining. Before we learn about treatment for COVID-19, walk us through Dyadic and the C-1 gene expression platform.
Sure. So the C-1 technology platform has been developed over the last 25 years to take DNA gene sequences, put them into a cell line, in this particular case, the fungal cell C1. It's a hyperproductive cell line, and it turns the gene sequence into a protein. In this particular case, it could be an antibody for a treatment to the COVID-19 virus, or it could be a vaccine, which would be a antigen. And so we've developed this for industrial biotech, licensed it to the biggest companies on the planet, for example, Shell Oil, BASF, Abengoa, and then DuPont purchased our industrial business on December 31st of 2015 for $75 million. And then we kept the rights to use this technology for both animal and human health. Because at Dyadic, in addition to the coronavirus outbreak, we're working on animal health products, vaccines, antibodies. We're working on human health vaccines and antibodies and metabolites and potentially even viral vectors. So most of our business doesn't relate to the coronavirus, but as it came up, we had an opportunity to help because we have this very powerful capability to make a lot of protein and obviously vaccines and antibodies of proteins at very, very large scale, at very low cost, in massive quantities. And so we're applying that technology to help break or sort of combat the outbreak today, be ready for the pandemic tomorrow, future pandemics, and to lower the cost of prescription drugs in the future.
And Mark, that's exactly it. It'll be great once treatment comes online, but then you also have the challenge of manufacturing in mass quantities, like you had said. But at the end of the day, and this is a hot button issue, it has to be accessible and affordable to everyone, because as you're very well aware of, pandemic, disease, viruses, it doesn't matter what your background is, political affiliation, gender, race, it doesn't pick on that basis. So I think that's really what the challenge is going to be, is the logistics behind it.
Yeah, and I agree with that. I think that people that are missing the point that there's two components to a vaccine. There's the sequence that encodes for the vaccine, for the antigen, for example, and there's a cell line that's used to produce the vaccine. And if they don't communicate with one another, the gene and the cell, your output is very low. And when you put a gene into an inefficient cell line, you get very small amounts of too expensive vaccines that aren't available in enough quantities to provide to the global population, let alone maybe even the American population. And C1 has the ability to do it all. It can decode the gene sequences. It can produce massive amounts very affordably at flexible commercial scales. And I think that's important to realize. Flexible commercial scales is we can grow in small fermenters a lot, and in big fermenters just a heck of a lot more.
So realistically, what would a time frame look like to bring it to market, assuming that all the approvals go through and there's no hiccups along the way? What's a realistic timeline?
Well, I think there's a different timeline for each aspect of this. And so, first somebody had to develop a gene sequence or a vaccine candidate that actually has the potential to be the vaccine, right? And so with that, we've already been working with the European Union, the Israeli Institute of Biological Research, Scripps in La Jolla, through UFOVAX. So we have 3 ongoing sort of vaccine candidates that have been developed and are ready to go prime time. 2 of those are already being put into our C1 cell, and we're seeing excellent results so far to date in terms of productivity. And then the next step, they have to go into animal models. And so the animal models, then they test them to make sure that they actually have the efficacy and the potency that you expect. And then you go into clinical stage, phase 1, phase 2 trials, and ultimately you get the EMEA, FDA, or other governmental regulatory approvals. And really for the recombinant protein approach, which is what we're dealing with, you're not going to see those vaccines, just as people like Sanofi have talked about in J&J, until the earliest, sometime in mid to late 2021. So we're all hoping that Moderna and Inovio, that their vaccines work because they can supply smaller amounts, maybe sooner, but it's still an open question on whether those vaccines and those technologies will actually give the immune response, create enough antibodies to actually break the virus down inside the body or neutralize it. And so we believe that in our case, we have a unique opportunity to provide billions of doses after the regulatory approval, make them rapidly, make them quickly, make them at low cost at flexible commercial scales. So you can actually produce, and our estimate is based on the results that we had in the ZAPI European program we were in for the last 5 years. And ZAPI is Zoonosis Anticipation Preparedness Initiative, which is exactly zoonotic diseases to humans. In that case, we worked on MERS expression in C1, SPV antigen in C1, and then through the animal studies, the SPV antigen and the productivity level of C1, which was 35 times greater than the baculovirus cell line that's being used by Sanofi, for example, Novavax. Our cell line is more productive, it's more robust, and it can provide a lower cost and higher volumes. So based on what we've seen, and in that particular case, we used a dose of 20 units per gram, we estimated that we can produce 10 million doses in a 200-liter fermenter in 121 hours of the antigen. Or if you put it in a 2,000-liter fermenter in 121 hours, which is only like 3 or 4 days, it would produce 100 million doses. If you run it in a 20,000-liter fermenter in 121 hours, then you can produce 1 billion doses. So you're talking about in a month, if you had multiple outlets, for example, you ran it in India, you ran it in China, you ran it in Brazil, you ran it in Europe, and you ran it in America, you could produce the world's needs of 7 billion doses of vaccine antigens in less than a month. And that's remarkable. And not only can we produce the dosages that are required, they can do them at a cost that'll be at a fraction of the cost on the upstream antigen production that you would get out of any other cell line that's out there today.
Yeah, that certainly is remarkable, Mark. Thank you so much for joining us on Trade Talks.
Thanks for having me.
All right, and thanks for joining me. I'm Jill Malandrino, Global Markets Reporter at Nasdaq.